Knowledge seekers, welcome to my channel. Grab your pen and paper. We're going to discuss the development of mammalian kidney, or what you can say the embryology of mammalian kidney. The very first thing that you need to keep in your minds is that kidney, mammalian kidney, was made by intermediate mesoderm. What is intermediate mesoderm? Intermediate mesoderm is one of the major lineages of amniotic mesoderm. What is its location? For that, I will refer to these slides provided by Kharkiv National Medical University. And here you can see the transverse section of the three-layered embryo towards the end of third week of development. And here, marked by number 2 is the intermediate mesoderm. This one here. This is, this is what we are talking about, this region, intermediate mesoderm. Now gets back to our textbook, which is Developmental Biology, 11th edition. So, development of kidney is quite an interesting process to learn. It is because the functional unit of kidney, nephron, contains more than 10,000 cells, and these cells are not of a single type, but there are at least 12 different cell types, and each cell type has some specific function, and it is located in a particular place in relation to others along the length of nephron. And it also amazing to know that each of kidney, let's talk about humans, each of human kidney has around 1 million of these functional units or nephrons. So now let's proceed towards the mammalian kidney development. The development of mammalian kidney progresses through three major stages. The first one is pronephros, then there is mesonephros, and then there is metanephros. I am repeating them: pronephros, mesonephros, and metanephros. The first two stages are transient stages. Only the third and final stage persists as a functional kidney. So we are going to discuss these stages one by one. What happens is that early in development, day 22 in humans and day 8 in mice, the pronephric duct arises in the intermediate mesoderm just ventral to the anterior somites. What are somites? Somites are segmental blocks of a mesodermal tissue that are formed from the paraxial mesoderm that is adjacent to the notochord. So the cells of this duct, the pronephric duct, migrate caudally, or you can say towards the tail end, and the anterior region of the duct induces the adjacent mesenchyme to form the pronephros, or the tubules of initial kidney. The pronephric tubules form functioning kidney in fishes and in amphibian larvae, but they are not believed to be active in amniotes. In mammals, the pronephric tubules and the anterior portion of pronephric duct degenerate, but the more caudal portion, or the portion that is present towards the tail end, as said earlier, caudal portion of the pronephric duct and its derivatives persists and serves as a central component of excretory system throughout development. This remaining duct is often referred to as the nephric duct or Wolffian duct. Let us take a look at it. This is the snapshot of same PDF that I have saved separately in order to not navigate from the through the PDF again and again. As we can observe that this pronephros, or the first stage, as the development progresses, gets degenerated. However, a portion of it remains, and this portion is called the nephric duct or Wolffian duct. So this is what we were talking about. Now let's get back to our text. As the pronephric tubules degenerate, the middle portion of nephric duct induces a new set of kidney tubules in the adjacent mesenchyme. This set of tubules constitutes the mesonephros, sometimes called the mesonephric kidney. Now look at here again. This is nephric duct and adjacent to it in the mesenchyme has been induced this mesonephros or mesonephric kidney. In some mammalian species, the mesonephros functions briefly in the urine filtration, but in mice and rats, it does not function as a working kidney. In humans, about 30 mesonephric tubules form these. Around 30 of these mesonephric tubules are formed beginning around day 25. As more tubules are induced caudally or towards the tail end, the anterior mesonephric tubules begin to regress through apoptosis. As we can see here, as the development progresses, the mesonephric tubules towards the caudal end or tail end are getting to degenerate or are regressing. Although it remains unknown whether human mesonephros actually filter blood and make urine, it definitely provides important developmental functions during its brief existence. The first of the function that mesonephros provides is that it is one of the main sources of hematopoietic stem cells which are necessary for blood cell development. The second function that mesonephros performs in mammals is that in male mammals, some of the mesonephric tubules persist to become the tubes that transport the sperm from testes to urethra. By those tubes, it means the epididymis and vas deferens. As we can see, that gonads are just present nearby to the mesonephros, so they are playing a role in development of epididymis and vas deferens. Then comes the permanent stage or the permanent kidney, the third stage, metanephros. The permanent kidney of amniotes, the metanephros, originates through a complex set of interactions between epithelial and mesenchymal components of intermediate mesoderm. In the first step, the kidney forming metanephric mesenchyme, which is also called as metanephrogenic mesenchyme, becomes committed in posterior regions of intermediate mesoderm, where it induces the formation of a branch from each of the paired nephric ducts. Let's get back to the diagram. Here is what is the metanephrogenic mesenchyme or metanephric mesenchyme, which is going to get developed into kidney. So what it does is that it becomes committed in posterior region of intermediate mesoderm and it induces the formation of a branch from each of the paired nephric ducts. These epithelial branches are called the uretic buds, and these buds eventually grow out from nephric duct to form the collecting ducts and ureters that take the urine, the bladder. Now take a look at this picture. This was the ureteric bud, this was the nephric bud, and this is what is going to happen. The branches are forming, and here one of the branches pointed as ureter. And what happens next is that when ureteric buds emerge from nephric duct, they enter the metanephric mesenchyme. The ureteric buds induce this mesenchymal tissue to condense around them and to differentiate into nephrons of mammalian kidneys. And as this mesenchyme begins to differentiate, it tells the ureteric buds to branch and grow. And these reciprocal inductions form the kidneys in mammals. As you can see in the diagram, it is the earlier stage. So in this stage, the mesenchyme is just going to get what you can say attracted to these branches. What we can say is that the metanephrogenic mesenchyme is condensing around these buds, the nephric buds. So this was all about the three stages of development of mammalian kidney. Now let's get a little bit of help from the slides. So the pronephros, we are talking about humans when we're we are referring to these slides. The pronephros is the simplest and most primitive kidney system, and it consists of 7 to 10 solid or tubular arranged cell groups in the cervical region, the head kidney, and it is gone by the end of fourth week. After that, the stage which comes is the mesonephros, and it is the intermediate and more advanced than pronephros. It appears during regression of pronephros and 10th to 26th somite level. Then comes metanephros, which is a permanent kidney, and it begins to develop early in 5th week and functions by 11th week. And remember, we are talking about humans in these slides. Here we have pronephros, or what we call the forekidney, which is a transitory structure as we had stated it earlier. So this is what it going to look like. Here at number 1 we have the nephrogenic cord, and at number 2 we have mesonephric duct, or and then we have 1 plus 2 mesonephrons, which are pointed out by number 2, and then we have the intestinal tubule and other structures which are not our concern in this lecture. This is one of the other diagrams. Here we have the mesonephric duct, and we have the mesonephric tubules, and then we have the ureteric bud, and then other structures that are not of our concern. And here we can see the degenerating pronephros on the caudal end. Then comes the mesonephros, and these tubules develop from the nephrogenic cord and opens into excretory or mesonephric duct, and they are gone by week 10 in females, and in males some tubule persist and become vas deferens as we studied in the textbook. Approximately 38 pairs of closed tubules S-shaped bend front internal glomerulus. Mesonephric duct develops laterally from the nephrogenic cord and extends from 8th smite to urogenital sinus. Here we have the mesonephros, or the second stage, which is also a transitory kidney. So here at number 1 we have the nephrogenic cord, and then we have the mesonephric duct at number 2, and 1 plus 2 are, these two are what we call the mesonephros. Here we have another diagram of it. We can see the number 9. Here we have number 9. This is the Wolffian duct, and at number 10 we have the mesonephric tubule. Here is an other of the diagram, but this is of the final stage, the third stage, the metanephros, and it is the definitive kidney in the 5th week of human embryonic development. Here we have at 1A we have pronephros, which is atrophying, or what we can say regressing, and 1B shows the mesonephros, which is also regressing, and this number 2 point out toward the Wolffian duct or mesonephric duct, and this number 3 point out toward the NC or nephrogenic cord, and then at number 4 we have ureter analog, and then at number 5 we have metanephric blastema, and then the other structures that are not of our concern in this lecture. Here is other diagram showing the metanephric outflow. As you can see, the numbers 2 point out toward the ureter analog, and number 3 point out metanephric blastema, and these 2 and 3 combined they form the metanephros. And number 4, here it is what is representing the mesonephric duct or Wolffian duct. Number 5, here it is the nephrogenic cord. Number 4 plus 5 combined are called mesonephros. 4 and 5, these two structures combined are called mesonephros. So metanephros and neurons or tubules develop from nephrogenic mass 26 to 28 smite level. They are located lateral to mesonephric duct. Internal dense layer which forms tubules or nephrons. Outer loose layer forms connective tissue capsule. System is derived from ureteric bud, which forms ureter, pelvis, and calyces collecting ducts, as we have studied shortly the textbook. Ureteric bud elongates and makes contact with nephrogenic mass, which surrounds bud like a cap, as we had said earlier in the textbook, that the metanephric mass or metanephric mesenchyme condenses around the bud. The tubules are closed, internal glomerulus, and migrate from pelvis to abdomen as the fetus grows. Blood supply from aorta changes as ascent occurs and becomes functional in second half. This metanephros or this functional kidney or the final kidney becomes functional in the second half of the pregnancy. Here in this diagram, we have ventral view of developing kidneys. The mesonephric tubules have been pulled to side so that they may be seen more easily. Here we have mesonephric ducts, and we have mesenchyme, and we have mesonephric tubules, and then we have ureteric bud, and then these are the parts. These at the caudal end are pronephros, then the mesonephros, and then the metanephros. So this is what is development of kidney in mammals, in particular in humans, and these are the three stages. The first two being transient, I am repeating again. Pronephros and mesonephros are transient kidneys, while the metanephros is the final and permanent kidney in mammals. And at the end of its development, this is what kidney looks like. Typical kidney that we might have seen at various places in diagrams, and its various structures are labeled. And this is the schematic cut through kidney sagittal section, and it shows various structures such as here we have ureter, and then we have major calyx, and then minor calyx, and renal pelvis, and collecting duct pointed out by number 5, and then metanephric vesicles and kidney lobe. And this is the enlargement of the inset nephron. Here we have various structures of a nephron. This was all about the development of kidneys in amniotes, to be specific in mammals and to be highly specific in humans. See you in the next lecture. I hope that this lecture proved beneficial for you, and for whatever the reason you were watching this lecture, have been able to get the desired content and in appreciable quality. So please like this video, subscribe to this channel, and do not forget to turn the notifications on in order to get the latest updates. See you in the next lecture. Have a nice day.